Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
So, good morning, everyone. I think now we can get started. So, welcome to the MIT Faculty Forum. I'm Jinxing Li. I'm currently a postdoc at Stanford University, and I will serve as today's moderator for the presentation. And the title will be the insulin pill that delivers. So, I'm currently postdoc working at Stanford University at Chemical Engineering Department, and I joined here 2 years ago. I work on drug delivery. I saw like a very similar kind of trajectory with Alex. Did PhD on drug delivery, and then my postdoc working on bioelectronics. I think the only difference is that he has a much better publication during his PhD study. So unfortunately, I also was selected by MIT Technical Review as the Innovators Under 35 last year. So today's broadcast being brought to you by the MIT Alumni Association and is sponsored part by the MIT Federal Credit Union, as well as MIT Professional Education and MIT Snow Executive Education. So as a reminder, we welcome your question during this chat. Alumni joining via Zoom can use the Q&A feature, which can be found on your toolbar. For those viewing on YouTube, you may add your question to the comment fields next to the stream. We also encourage you to tweet using MIT Better Word. We'll get as many questions as we can. So I'm delighted to introduce our speaker today, Dr. Alex Abramson. Alex is a postdoc fellow at Stanford University working with Professor Janan Bao, and he's currently developing ingestible and implantable device for a variety of therapeutic applications. And we are in the same group, and he's doing really exciting research. And recently, he just got the NIH postdoc award to support his research at Stanford, which is really exciting. And he received his PhD in chemical engineering at MIT, studying under Professor Rob Langer, where he worked closely with Novo Nordisk pharmaceutical companies and gastroenterologist Giovanni Travaso to develop commercially viable and safe pills for effective delivery of macromolecular drugs. So his work is published in journals such as Science, Nature Medicine, and has received considerable press coverage from such news outlets such as New York Times, New England Journal of Medicine, Wired, and so on. And he's also involved in the public health sector and has performed research on quantifying the quality of life impact that novel biomedical techniques have on patients and worldwide. So if you want to get more information on Alex, you can go to his own website, which is www.agabramson.com. So is his first name and A, and his I think and then his last name. Sorry. So yeah, and outside lab, Alex likes to play basketball and making bad jokes. So with that, I will turn the scene over to Alex.
All right, thank you, Jinxing, for that introduction. Jinxing also does some really great work in nanotechnology, and I encourage you to look at what he did and go to the MIT Technology Review website. He has some videos also from Stanford that really go over his work really well. So today I'm going to be talking to you about ingestible capsules for therapeutic injections in the gastrointestinal tract, specifically for the delivery of macromolecule drugs. And for those of you who have ever been prescribed a macromolecule drug, such as insulin or adalimumab, or who have ever taken a vaccine, you know that currently there's no pills for these medications. And the reason that there's no pills is the fact that the delivery through the gastrointestinal tract is extremely difficult. And this has actually been going on, this quest for oral macromolecule delivery has actually been going on for about 100 years. But before I get into my talk, I want to just give my disclosures. And so this quest has actually been called the quest for the holy grail of drug delivery. And it's not just me who's been calling this quest the quest for the holy grail of drug delivery. Two very famous professors in the field, Professor Dave Putnam at Cornell and Professor Nicholas Pepys at UT Austin, see this as a holy grail of drug delivery because the quest has been going on for about 100 years, ever since the purification of insulin in 1922 by Frederick Banting. About a couple months later, a professor published the first oral delivery of insulin in 1923 in the British Medical Journal. And in that study, he tried to deliver insulin using alcohol solution, and he saw that only 1 in 5 patients were actually able to see a blood glucose lowering effect. He saw extreme variability between the patients, and he saw an extremely low uptake of the insulin. So less than 1% of the insulin was actually getting into the body in these patients. But I want to first, give you guys a perspective and really talk about my perspective of why I think that this is called the Holy Grail. So imagine that you're one of the Knights of the Round Table and you're searching for the Holy Grail, and you finally, you come upon the secret to eternal life. But instead of there being a chalice in this cave to deliver the holy water, instead it's a syringe, right? I think that all of us would be pretty shocked to see that and maybe a little bit discomforted. And that kind of gives you an idea of what these patients who are taking these macromolecule drugs have to go through every day. So diffusion and degradation limits macromolecule uptake in the gastrointestinal tract. Insulin, unlike a pill like Advil, is a much larger molecule. It's about 100 to 1,000 times larger, different macromolecules compared to these small molecule drugs like ibuprofen. And for that reason, it's very difficult to pass through the gastrointestinal wall and be taken up into the bloodstream. Additionally, because insulin and other macromolecule drugs are proteins and nucleic acids, they're degraded by enzymes in the gastrointestinal tract. As these molecules are trying to diffuse through into the tissue wall, additionally, they're being degraded by the enzymes present in the stomach and the small intestine, making it very difficult to have a lot of absorption into the bloodstream. So people for 100 years have tried things like delivering nanoparticles, delivering mucus adhesive patches, and most recently, they've tried delivering permeation enhancers. And in fact, Novo Nordisk has recently actually had FDA approval for the first tablet that can deliver a oral biologic diabetes drug. This drug is called semaglutide, and some of you may have actually seen the semaglutide tablet being advertised on TV. But one of the key differences between semaglutide and a molecule like insulin is that the dose for semaglutide is extremely low. In fact, the weekly dose for semaglutide is anywhere between 0.5 milligram and 1 milligram per week when it's injected. But when it needs to be delivered orally, the delivery of semaglutide has to be between 100 milligrams to 280 milligrams per week to see the exact same therapeutic effect. This is extremely important when you think about drugs with even higher doses. For example, if you think about a drug like adalimumab, which needs to be dosed at 40 milligrams every 2 weeks, then you're talking about a 100x increase on that, which is significantly more than 100 milligrams. You're talking about 2 orders of magnitude larger. So even though this permeation enhancer is great for delivering drugs that have a very small amount of drug necessary to reach their therapeutic effect, when you talk about most biomacromolecule drugs, you need to come up with a different solution that's able to deliver a higher percentage of the drug that's loaded into the pill. Something that gastroenterologists have been looking at for a really long time is delivering local injections of drug to the gastrointestinal tract. So anybody who's ever had a colonoscopy may have heard about something called the Carlock needle. And this is a 25 gauge needle that's 5 millimeters long. And it's used to inject saline into the gastrointestinal tract whenever a doctor is removing a polyp. So the saline is injected underneath the endothelial tissue of the gastrointestinal tract, which raises up the polyp and allows the doctor to more easily remove it. But in addition to saline, there's also a little tiny bit of epinephrine in this solution. And the epinephrine has actually been seen to have a systemic effect because patients often have their heart rate increase whenever they're delivered this solution, which gave the idea that injections in the gastrointestinal tract probably have a very high delivery efficiency. Additionally, when they're delivering the injection to the gastrointestinal tract, no painful sensations are felt, and that's because the GI tract doesn't have any sensory receptors that sense pain, specifically in the stomach and the intestines. Additionally, the gastrointestinal tract has very rapid tissue regeneration. Anybody who's done a little bit of studying of the stomach knows that the tissue wall regenerates every 1 to 2 days in the stomach, and about that same frequency in the small intestine and the colon as well. But this local delivery of drugs using a Carlock needle and an endoscope is great, but how do we actually deliver a macromolecule drug via an injection using a pill? And so that's what I'm going to talk about today. But when you think about the gastrointestinal tract, one of the first questions is, where do we deliver? The gastrointestinal tract has several organs, and I'm going to go through each one and talk to you about the advantages and the disadvantages. First up, we have the esophagus. And the esophagus is a very difficult place to deliver an injection because it has a very short passage time of about 5 seconds to a minute. And additionally, those of you who've had those of you who have ever had swallowed a chicken bone would know that delivering any sort of injection to the esophagus may cause some pain. Swallowing a chicken bone is very painful, and so it could be difficult to deliver an injection to that area. The stomach, on the other hand, has the first area of the gastrointestinal tract where a pill would actually be able to sit for an extended period of time. The passage time in the stomach is anywhere between 1 to 4 hours, allowing us to deliver an actuation mechanism that's able to specifically deliver to the stomach area. And so the first pill I'm going to be talking about today is called the self-orienting millimeter scale applicator, or SOMA for short, and that was published in Science last year. I'm the first author on the paper, and my advisors, Professor Robert Langer and Giovanni Traverso, are the senior authors, but there are also quite a few other people who worked on that, and I'll acknowledge them throughout. The other area that a lot of people think about delivering macromolecule drugs is the small intestine. And in fact, the vast majority of the work for delivery of oral macromolecules has been in the small intestine area. And that's because the small intestine has a very large vasculature system. And when you think about the absorption of nutrients, most people think about that through the small intestine. But one of the issues of the small intestine compared to the stomach is that it has a very thin tissue wall. So if you're delivering an injection to the small intestine, you have to worry that you might cause a perforation. The small intestine has a tissue wall of about 1 to 2 millimeters thick, whereas the stomach has a tissue wall thickness of about 4 to 6 millimeters. So in order to solve that, we created something called the luminal unfolding microneedle injector. And so compared to the needles that we use in the SOMA system, which are in the millimeter scale, the LUMI system uses much, much smaller microneedles that are on the sub-millimeter scale. And so I developed this along with my co-first author, Esther Caffarel, and my advisors, Bob and Gio, as well as several other people. And this was published in Nature Medicine also in 2019. And I wanted to give you guys, before I go into the details of the pills, just some histology of what it looks like when you inject using these pills. And so what you see is that we're delivering into the mucosa and actually through the mucosa in terms of the soma, just to this area called the submucosal layer where the vasculature begins. With the luminal unfolding microneedle injector, because we're so worried about perforation using these injection needles, we're only delivering to the mucosa area. There's also several other people working in this space. I specifically want to highlight Rani Therapeutics, which is using a pill for the delivery of drugs into the small intestine. And they actually ran a phase 1 clinical trial and had a very positive readout. But there are several advantages to the pill that I want to highlight. One of them is that it reduces the stigma associated with the delivery of these drugs, and it improves patient quality of life. I think that that's pretty self-explanatory, but I wanted to give you guys a fact from the public health sector. For people suffering from diabetes, patients actually experience 10 times greater discomfort when they are delivering an injection versus when they're taking a pill to treat their disease. Additionally, it eliminates the biohazardous needle waste. These pills don't have any solid needles in them. The needles themselves are made of the drug, and therefore they dissolve, and by the time they pass out of the body, there's no biohazardous needle waste to dispose of. Additionally, these pills are replacing very large auto-injectors, and so we reduce the plastic waste footprint by greater than 90% if you're replacing this pill with an auto injector. And lastly, because these pills use solid formulations rather than liquid formulations that are used in the auto injectors, it enables a chemical stability in high temperature environments, meaning that you could put these pills in your pocket, walk around with them for 4 months, and have them still be stable after that period of time. So I want to talk about the self-orienting millimeter scale applicator first, and then I'm going to talk very briefly about the luminal unfolding microneedle injector. So I've been talking a lot about this concept of self-orienting, and why is that important in the stomach? Well, if you think about the stomach is a very large organ with a very wide space. And if you were delivering a needle into the stomach with a pill and you didn't have a preferred orientation, it's almost a 75% chance that that needle is going to inject into the lumen of the stomach rather than into the tissue. And if it injected into the gastric fluid, that would mean that the drug would degrade very quickly, and it wouldn't also be able to penetrate through into the tissue wall and be absorbed systemically. So the idea of this self-orienting millimeter scale applicator is that it localizes within a little bit less than a second. Within the order of minutes, it injects its drug into the tissue, and within the order of about an hour, the drug dissolves and allows for uptake into the body. How does this device self-orient? Well, it uses a specific shape and mass distribution where the center of mass actually only has one local and global minimum. If you guys have ever played with a Weeble Wobble toy or anything along those lines, you know that Weeble Wobble toys, when you push them over, they reorient themselves, and that's because their center of mass is very low, but additionally, the shape of the device is set up so that it only has one stable configuration. Additionally, I'm going to talk a little bit about how this device delivers a safe and efficacious actuation and how it's able to deliver a clinically relevant dose. But first, I'm going to go more into the self-orienting. So while many people think about the Weeble Wobble toy in terms of self-orienting, the first thing that I thought of in terms of self-orienting is the leopard tortoise. And in fact, this device is inspired by the shape of the leopard tortoise. And it's able to self-orient with 100% efficiency because we're using physical properties of the device. The upper portion of the tortoise has a very pointed top, whereas the lower portion of the tortoise has a flat bottom. This is extremely important to the soma system because we want to destabilize the upper portion of the system, whereas we want to stabilize the bottom portion of the system. Once the soma has oriented in the stomach, it's actually very difficult for it to get pushed over, unlike the Weeble Wobble toy. And that's because it has that corner at the bottom of the soma and the flat bottom that makes it difficult to push over from its preferred configuration. And that's very similar to the leopard tortoise that doesn't want to be pushed over by any sort of predators. When it is flipped over in its unpreferred configuration, though, within about 100 milliseconds, the soma is able to reorient itself and extremely quickly and consistently. So now that we've talked about that self-orienting aspect, I want to talk a little bit about the actuation mechanism. And so in order to actuate this system, we use a combination of a compressed spring and a sugar glass material. Sugar glass is extremely brittle, and we want to use a brittle material compared to a ductile material because we want the energy from the compressed spring to release near instantaneously. We're also using a compressed spring because the compressed spring allows for uniaxial release of the energy, and that's what we want. We don't want the energy spraying all over. We want it specifically pushing the needle down into the tissue. Instead of using a sugar, if you used a taffy, a ductile material, the spring would release very slowly, which wouldn't give us the momentum to allow for the injection of the drug into the tissue wall. We did several models, and then we compared them to experiments to see how coating the spring in sugar glass would change the time that the actuation would occur. And so as we increased the thickness of the sugar glass around the spring, as you would expect, we also increased the time for the actuation event. And so based on the thickness of the sugar glass that we put, we were able to time the actuation event to occur within 5 minutes, allowing the device to pass through the esophagus, land in the small intestine, self-orient, and have about a minute or so before the drug was actually injected into the body. And so here I have a picture of the spring releasing from the sugar glass. And this is filmed at 10,000 frames per second and slowed down by about 3,000 times. So this spring is completely releasing all of its energy within less than 1 millisecond. And so I wanted to show you what that looks like from the outside of the device. We have a little bit of water that's actuating the spring from the inside. And this device is actuating into porcine stomach tissue. When the spring releases, all of the water sprays out. And this is again in slow motion. When the injection occurs, we want to make sure that we're not perforating the tissue, but we also want to make sure that we're delivering to the area that is vascularized in the stomach. And so, as I mentioned before, with this Carlock needle system, we wanted to emulate exactly what was occurring with gastroenterologists and what they were using previously to make sure that our device was safe. The Carlock needle is actually able to deliver into the submucosa area of the gastrointestinal tissue. And so the submucosa, as you can see from this area on the left, this animation on the left, is really where most of the vasculature is in the stomach tissue, allowing for a rapid uptake of the drug in the stomach and then delivering that to the rest of the body. When we delivered our insulin post, which you can see on the bottom right-hand side of the screen, the insulin is actually specifically in the tip portion of the post that we're delivering. And the rest of the post is made of a biodegradable shaft, meaning that all of the drug that we're delivering is able to actually enter the tissue and none of it is degraded by the gastric fluid in the stomach. And so what we see is that we're passing through the mucosa and reaching that submucosa area. You can also see the smooth muscle stained histology on the far right-hand side of this slide. To look and make sure that we were actually delivering with quite a bit of cushion, meaning that we weren't getting very close to the point where we would potentially be perforating the tissue. We also delivered a post using 9 newtons of force. And this wasn't an insulin post, it was a post made of stainless steel, meaning that the speed of the post at its point of delivery was going to be higher and the mass of the post was going to be higher, meaning that the momentum would be significantly higher. And what we saw was that even if we delivered using the stainless steel post and using 9 newtons of force, that we still were not able to perforate the stomach tissue. The muscle tissue of the stomach is actually very difficult to perforate because it's extremely tough, whereas the mucosa and the submucosa is much more easily penetrated. And so this makes it easy for us to deliver the insulin or any other macromolecule drug specifically to that submucosa area due to the changes in the mechanical properties of the tissue. And so this is what I think is the most exciting slide right here. And it's the uptake of the insulin over time. And so we used insulin as a model drug in this specific study, but insulin is really not only it's not the only drug that can be delivered. Any drug can be delivered. Macromolecule drug like adalimumab, monoclonal antibodies, nucleic acids could be delivered using this system. Proteins and peptide drugs can be delivered using this system, and even small molecule drugs that aren't able to normally be absorbed very readily in the gastrointestinal tract. And so what we saw was that compared to a subcutaneous injection of the same micropost and the injection using the Soma system and the manual injection of our post into the stomach through a surgical procedure, we saw a very similar amount of uptake if we looked at the area under the curve over the course of about 3.5 hours. And that we compared to if we delivered the post using the Soma system, but the Soma actually didn't inject the drug into the tissue wall. So we saw a localization of the drug, but no injection. And so this was able to show us that the injection is what actually was causing the delivery and the uptake of the drug, not the localization to the tissue wall. One of the benefits of using insulin as a model drug is that you're able to see a blood glucose change very rapidly. And so immediately, while you're doing this in vivo experiment, you're able to see a readout that is positive or negative, depending on whether or not your system is working. And so we saw actually a very similar blood glucose change between the subcutaneous injection, the manual stomach injection, and the injection using the Soma pill for the system. And those were statistically significantly different than if we delivered no injection at all of the insulin. Also, I mentioned that because we're using a solid dosage form, these formulations in the Soma system are stable. And so 2 of the things that we look at are the high molecular weight protein percentage and also the purity of the insulin in the system. And so in that micropost that we're using, we're actually able to see 16 weeks of stability of the system. Usually you're looking for a purity of greater than 80% and a high molecular weight protein concentration of less than 5%. And so compared to a solution, which is in red, you can see that the stability of our posts, even at 40 degrees Celsius and 75% humidity, is still very high over the course of 16 weeks. And I mentioned that we can use lots of other different molecules in the system. And I want to just mention a couple of simple enzymes that we tested in our post formulation. We showed that both lysozyme and glucose 6-phosphate dehydrogenase were able to maintain their enzymatic activity even after compression into the micropost formulation. One of the things that's very important when you're delivering a capsule into the gastrointestinal tract, especially if this capsule is made of non-degradable components, like the shell of our soma system and the spring, which remains inside of the capsule after actuation, we want to look at the size. So some of you guys might be familiar with the endoscope capsule, the PillCam system, which is used as an alternative to a colonoscopy or an upper endoscopy, or it's used in addition to that if there's potential problems in the small intestine. And this pill is actually extremely large. It's about 13 millimeters in diameter and 30 millimeters long. And so when you're delivering maybe once every 10 years, when a patient needs an endoscopy, that might be considered okay, even though the pooled retention rate for this capsule is 1.4%, which is extremely high. But if you're thinking about delivering a pill every single day, a pooled retention rate of about 1.4% is much too large. That would mean that patients would be going to the hospital for what's considered a medical emergency. Significantly, often maybe you have 1 in 100 patients who's prescribed this drug having to go to the hospital for a medical emergency. And so when we designed this pill, we actually designed it with a different type of non-degradable system in mind, and that's called the Oro system. It's an osmotic pump capsule that's used to deliver drugs like Concerta and Procardia, and it's significantly smaller. At its maximum, it's only 12 millimeters in diameter for some of the systems and 15 millimeters long. Actually, the Concerta system is 9 millimeters in diameter and 15 millimeters in length. And the Procardia capsule is more of a tablet, and it's 12 millimeters in diameter all the way around, and it's 4 millimeters long. The Soma system is smaller in volume than both of those systems, meaning that we expect that when it's dosed in very large quantities to a large population of patients, that we would have an extremely low obstruction rate case compared to what the Oro system is seeing of about 1 in 29 million. And the Oro system has been approved by the FDA for daily dosing. So of course, we can't test our pill in 10,000 patients quite yet. We're still in preclinical studies. But what we did instead was we delivered a week's worth of Soma pills to a swine. And we watched over time as the swine was able to pass all of those pills. Swine have a much longer passage time for the gastrointestinal tract. If you think about gastric emptying time for a swine, it's anywhere between 24 and 48 hours just to pass through the stomach, whereas a human takes about 1 to 4 hours. And so although it took a little bit over a week for the devices to pass in swine, we expect that it would take on the order of 2 days to pass through a human. And that just is in relation between the human and the swine gastric emptying times and gastric transit times. Now I want to talk a little bit about the luminal unfolding microneedle injector as well, just very quickly. So this one targets the small intestine. And it's important to think about devices that target both the stomach and the small intestine, because the small intestine has specific receptors in it and molecules that are unique to that area. And so you might want to deliver specific drugs to the small intestine, even though it's not quite as safe as delivery to the stomach wall, to the stomach's much thicker tissue wall. So the Lumi, the way it works is that it's kept inside of this capsule, which doesn't begin to actuate until it passes through the stomach. It's pH actuated, and the stomach has a very low pH of around 1 to 4, whereas the duodenum has a pH of about 5 to 6. And so this enteric coating begins to dissolve once it enters into the small intestine, and water begins to enter into the bottom chamber of this capsule. There's a spring inside of that capsule, which is encapsulated again in either a sugar coating or a polymer coating. And as that polymer coating begins to dissolve, the spring actuates after that period of time that it takes for that coating to dissolve, and the device is propelled from the capsule. And the device actually contains an elastomer in its core, which allows the 3 arms of the device to push outwards towards the small intestine walls and deliver very tiny microneedles into the mucosa of the small intestine. And while we needed to deliver to the submucosa of the stomach, because the small intestine has much thinner walls, and it also has a much larger vasculature system, even when we deliver to the mucosa, we see very rapid uptake. And I'll show that a little bit later in the slides. So this is a picture of the Lumi device. You can see that we have the spring system on the far left, then we have the core system in the middle and the arms on the far right. And again, this system is very small. It's actually closer when it's in its full capacity. It's about 30 millimeters long and 9 millimeters in diameter. But after it's actuated, it's able to break up into systems that are smaller than the Oro system, so smaller than 9 millimeters in diameter and 15 millimeters in length. And we took an X-ray picture just to show what it looks like when the device is inside of its capsule in the swine and when it's deployed. We put a couple of metallic pieces in the arms for this picture, but the metallic pieces aren't present when we're delivering the capsule and not taking pictures via X-ray. And so something that was important to us was to be able to change the capsule release time and vary it to be able to target different areas of the small intestine. Whereas with the Soma system, because the stomach isn't extremely large, it may be delivering to the same area of the stomach every time, potentially causing chronic issues after the delivery over the course of 6 months, a year, 2 years. With the Lumi system, we can deliver to different areas of the small intestine by varying the capsule release time. And so by varying the encapsulation of the spring in different molecular weights of polyethylene glycol, we're able to change the capsule release time anywhere between an hour all the way up to 5 hours. And so there were multiple techniques that allowed us to confirm the needle insertion using the Lumi system. The first thing that we did was we looked at just the mechanical characterization of the tissue, and we compared swine tissue to human small intestine tissue. And we saw as they had very similar characteristics. And we also looked at what the force was to perforate the tissue, because we didn't want to perforate the small intestine tissue. And so what we were always doing was delivering a force per needle that was less than the force required for perforation. And so we used two different techniques, histology and optical coherence tomography, to look and make sure that when the Lumi system was released inside of the small intestine tissue, that those microneedles that I was talking about were able to deliver into the tissue. The microneedles themselves are about 400 microns in diameter and about 900 microns long. And so they're thinner themselves than the thickness of the small intestine tissue. And again, what we saw was that if we delivered the Lumi systems, we were able to get a uptake of the drug, and we were also able to see a blood glucose lowering effect. We weren't able to see quite the same percentage of drug delivered via the Lumi system as compared to a subcutaneous injection of the same formulation. And that's because the Lumi system, the microneedles themselves were not able to penetrate fully into the tissue. Likely only the tips of those microneedles were going in. And so we saw about a 10% delivery efficiency compared to the subcutaneous injection of those same dissolved microneedles. We also delivered a microneedle patch directly to the intestine manually. And when we did that, we were able to see almost an equivalent uptake compared to the subcutaneous injection of dissolved microneedles. And so what that might suggest is that we need to deliver a Lumi system that has a larger force to allow the microneedles to penetrate further, or the way that the Lumi system is opening, maybe not all of the microneedles are inserting to their full length. But we are able to see a significant uptake of the insulin, and it's actually much larger compared to almost all of the different systems that are out there right now in preclinical and clinical trials. So I want to give a couple of ideas looking forward, right? Because these systems are still in clinical trials. We expect them to go into clinical trials within the next year or two, but there's still some work that needs to be done. And so the first thing is just talking about other drugs. Insulin itself may not be the best drug to go to market with for these systems. And that's because insulin has a very tight therapeutic window and it requires a very exact dose and it has a very short half-life. What we're looking for in terms of an optimal drug for oral delivery, especially as the first candidate for proving this system out in humans, is a drug with a wide therapeutic window and a long half-life. Meaning that, let's say there's a 99% chance that this is going to deliver. Even if there's a 1% chance and that one day you don't get the delivery of a drug, you're still able to keep the concentration of drug in your system up at a therapeutic level. And so when we talk about those kinds of drugs, adalimumab obviously comes to mind because it's the largest selling drug in the US. It's a monoclonal antibody and it's dosed at about, I think 50 milligrams every 2 weeks or about 25 milligrams per week. But if you compare that to delivery using the permeation enhancers, like how Novo Nordisk is delivering their semaglutide, if we had to increase that dose by 100 times, right, we're talking about grams of drug which become actually too expensive to deliver. Many people think about the idea that pharmaceutical companies are really, really overpricing their drugs compared to how much it costs to create the drug. And yes, that is in fact true, right? It costs anywhere between $25 to $100 per gram to produce drugs. But when a pharmaceutical company is selling you a drug, even if they're selling it at a 100 times markup compared to how much it costs them to deliver or to create that drug, or a 1,000 times markup, if you're talking about delivering 100 times more drug, then you're talking about a 100 times lower profit margin compared to what you were talking about originally. Maybe you're making 99.9% profit if it only costs 0.1% of the cost of your drug to produce that pill. But if you multiply that by 100 times, suddenly 10% of your cost isn't just raw materials alone. And in addition to that, you need to pay for the permeation enhancer, which can cost approximately the same amount as the drug. It needs to be delivered at a 10 times higher dose than the drug itself. So when we look at the actual necessity for getting a willingness to pay threshold for patients, what we're looking at to deliver something like adalimumab is somewhere around a 10% bioavailability necessary. And so this is from a study that I performed in collaboration with the Harvard School of Public Health and the Brigham and Women's Hospital, and is published in the Journal of Pharmaceutical Sciences. If anybody is interested in that, I'm somewhat proud, because it's not something that engineers normally do, is look at the willingness to pay thresholds for their systems. But I see that I'm getting some questions on that, and I'd be happy to answer the questions in the question and answer session. Another thing that's important to look at is mitigating food effects. When you talk about the injection into the tissue wall, you want to make sure that the tissue wall is clean. And so right now we've tested this only in fasted state animals, but we've been developing these membrane systems to allow the drug post to penetrate in, but not allow the food to penetrate into the pill. And so what we want to make sure of is that our system is not clogging up with food, but additionally that we have a clear path from the device into the tissue at all times. And so we've been working at different ways to mitigate the food effects in that way. Looking at new drug formulations is also important. I mentioned semaglutide and adalimumab, but also small molecules like epinephrine. And then the nucleic acids has actually had a significant amount of press just in the past day, because we've seen that the coronavirus might have an mRNA vaccine that could potentially be used. And so delivering these nucleic acids is really the next generation of drugs, and hopefully we'll be able to deliver those drugs orally using this type of system. And lastly, I've mentioned this a little bit before, but identifying the potential chronic effects. We know that if you're injecting into the same location subcutaneously every single day, that could not be good for the body. There might be a large amount of localization of the drug in that area. And additionally, you could get effects just from the delivery of the injection itself. And so rotating the injection site around the body when you're injecting subcutaneously is important. And we need to see how important that is when you're injecting in the gastrointestinal tract. As we said about the Lumi system, the capsule itself can change where it's delivering based on the time that it's able to actuate. But the Soma system might be delivering to the same area of the stomach tissue all the time. The stomach tissue, of course, has the ability to regenerate fairly quickly. And so that may not make a difference. In the acute studies that we looked at, when we delivered the system and then we pulled out the system and we took histology, we actually weren't able to see the hole where our needle or post was injected into in these systems within about an hour after the delivery. And that's because the mucus is very fluid in the stomach and the small intestine, covering up that hole. And also the mucosal layer itself is somewhat fluid as well and able to reorganize itself very quickly. But it's still important to look at the potential chronic effects of delivery of the dose into the same location for multiple months at a time. So I just want to give a quick overview. The device, the Soma device, is able to localize, inject, and dissolve. Same thing with the Lumi, just using a slightly different tactic. But those are the three main things, right? We need to localize the system to the tissue wall. We need to inject the drug, and then the drug needs to dissolve and be taken out into the body. But the things that I want to bring to your attention is that orally dosed devices can yield significant biologic drug uptake. Compared to the standard of care right now, which is the 1% bioavailability with the permeation enhancer that Novo Nordisk has come up with, we're talking about orders of magnitude greater to biologic uptake, potentially allowing for other drugs besides semaglutide to be delivered. These systems deliver the doses reliably and precisely, and that's because they're using physical methods of drug delivery. We don't need to worry about any variability associated with diffusion or any sort of chemical reactions going on. The self-orienting system is using this center of mass to ensure that it's always going to localize to the tissue wall appropriately. And lastly, that the Soma and Lumi can potentially be cost-effective platforms for the delivery of multiple biologic drugs. Insulin we used here as a model drug, but again, I want to emphasize that just because it was used as the model drug doesn't make it the best drug for the system. It just makes it the best drug for the preclinical trials. So there are a lot of people that I want to acknowledge. There's quite a few people from the Langer Lab, formulation scientists, undergraduates, electrical engineers and mechanical engineers, technicians and the animal technicians. So people helped us do the OCT. As I mentioned, I collaborated at the Harvard School of Public Health and the Brigham and Women's Hospital. But also a lot of people at Novo Nordisk were actually intimately involved in this research. And so all of these people who are listed here actually helped perform the research, either as animal technicians, formulation scientists, mechanical engineers. And without them, we wouldn't have been able to get this pill to where it is today. And so on the right-hand side, you can see a picture of the Langer Lab. It's actually very large. I don't know if any of you guys as alumni were able to be part of the Langer Lab at any time, but you may be wondering where I am in that picture. And so I have a arrow that I think just popped up. That's me in the orange shirt right there. And so every year we would take a nice picture in front of Bob's Beach. The lower left-hand picture is a picture of some MIT people and Novo Nordisk people during one of our collaborative visits. And in the bottom right-hand side is a picture of me and my new group at Stanford in the POW group. And Jinxing, I believe, is actually in that picture as well. And with that, I'll wrap up, and I'm happy to take any questions. And thank you guys for listening today.
Okay, thanks, Alex, for the great talk. I think I got a few questions for you. The first one is regarding the bioavailability. So for macromoleculars, if the bioavailability is very poor, so what will happen with when they go through the intestines? Will they be just pass through your feces, or will they be absorbed any elsewhere? So is there any problem associated with that? Yeah.
Sure. Yeah. So as I mentioned, right, not only is the low bioavailability an issue, but the variability is also an issue from patient to patient. So what's really bad is that if you just took a pill of solid insulin, in most patients, there would be no uptake. Whereas, let me get my camera back on, sorry. In most patients, there would be no uptake, but maybe in one patient, you would see significant uptake, which could be very dangerous. But traditionally, the enzymes in the gastrointestinal tract are going to degrade almost everything. And the tight junctions between the tissue wall won't allow any macromolecules through. And so you'll see about 0 to 0.1% uptake if you don't have any sort of permeation enhancer.
Mm-hmm. So the second question is about the method of administration. So can you compare the first pass metabolism via GI absorption to the port vein versus directly like injection to the circulation?
Sure. So there's a couple of different things that I can compare to. One is I can compare to an IV injection, and another one is that I can compare to a subcutaneous injection. Not all macromolecule drugs have a 100% bioavailability when they're subcutaneously injected. For example, recombinant human insulin has an absolute bioavailability when injected subcutaneously of about 50%. And so when you're looking at the absolute bioavailability compared just between a subcutaneous and an IV injection, you're going to see a difference. Semaglutide, on the other hand, has an absolute bioavailability when injected of about 80 to 90%. And so that one is able to more readily enter the bloodstream. And that might be due to the longer half-life of the drug. Insulin is degraded very quickly, even in the subcutaneous space. But when you're looking at the delivery into the gastrointestinal tract, something that you have to think about is the first pass metabolism through the liver, as was mentioned in this question. Specifically, the small intestine is getting delivered through the portal vein into the body, and so that's passing through the liver. When you're talking about delivery to the stomach, you are not necessarily getting a first pass metabolism through the liver and absorption into the portal vein. The stomach has, depending on where you're injecting into, different drainage points into different vasculature systems. And so what we've seen is actually delivery into the stomach is not causing necessarily portal vein delivery. It could be causing just traditional systemic delivery.
Mm-hmm. Third one is when you talk about the diabetes, are you talking about the type 1 or which type, subtype?
Yeah. So it's probably best for type 2 diabetics. Type 2 diabetics are taking large doses of bolus insulin at the beginning of the day. This is basal insulin that is long lasting. And that would probably be the preferred method of delivery for this particular insulin. When you talk about fast acting insulin, you have the issue of having to deliver a very specific dose, which could be done using this capsule, but you need to have different capsules with different doses. For example, you'd have a 4-unit capsule, an 8-unit capsule, and a 12-unit capsule, very similar to what Afrezza, the inhalable insulin, has for their packets. But when you're delivering a large amount of insulin that lasts for a long period of time, you'd be able to do that in the morning before a patient has had any food on an empty stomach. And so that would be much easier to deliver, and that's more used in type 2 patients.
Mm-hmm. The next question is about the commercialization, and specifically he or she asked if there's any Japanese company interested in commercialize this technique.
So MIT and Novo Nordisk worked very closely together on this system, and currently MIT and Novo Nordisk are working to commercialize this worldwide. I don't know about any sort of local areas, but I do know that we have quite a few patents that have been filed internationally in many different countries around the world. And so that's the plan for commercialization right now. And unfortunately, I can't speak any more about it because I don't know what's going on in the commercialization efforts myself.
The next one is about the long-term effect if you do multiple injections in the digestive tract. So what will happen?
Yeah, so I mentioned that a little bit towards the end. And the answer is right now, we don't know. We haven't run those chronic studies. We have dosed multiple devices to a single animal. And then we also have looked at the tissue after 1 day, after 1 week. And then we looked at the entire gastrointestinal tract as well to see if there's been any effects after the acute administration of the system and passage of the pill. And we haven't seen anything that would show any sort of potential side effects from this injection. But that being said, we haven't done the chronic studies.
Mm-hmm. The next one is how is the NUMI going to work with large number of patients using PPI, I think the proton pump inhibitor that neutralize the gastric pH?
Yeah, that's an interesting question, and it's something that we've been thinking about. Not every pill is going to work with every patient. And so when you're thinking about what pills should be prescribed, the physician should think about what the side effects would be. There are quite a few pills out there that use a similar pH mechanism. And so those pills need to be prescribed only to patients who are able to take them.
Mm-hmm. So the next question is, how does the kind of dosage adjust to different like blood glucose level? So would you need different dose tiers, or how do you do that?
So different dose pills is probably the best way to go about it. However, there is some really interesting research being done in Zheng Gu's lab at UCLA that's looking at creating needles that are glucose responsive insulin. And so I'm not personally working on that research, but I think it's very cool and I've been following it very closely. And so if you're able to deliver a post that is filled with a glucose responsive insulin formulation, you could theoretically deliver a large amount and then have the chemistry allow for the slow delivery or the sustained delivery over time.
Mm-hmm. Yeah, that's also what I noticed about. So the next is that what is the mechanism that draws the insulin through the needle after the injection?
What is the mechanism that draws the insulin through the, can you repeat that question, please?
Yeah, I think, yeah. What is the mechanism that draws the insulin through the needle? I think it probably, how do you, after injection, yeah, he's asking.
Okay, yeah. So we're actually using solid needles here, where the insulin is concentrated at the tip portion. So we don't actually have a liquid formulation being pushed through the needle. The needle itself is the insulin. And so because of that, we don't have any sort of needle that is sticking out that doesn't dissolve as the device passes through the gastrointestinal tract. And additionally, we don't need to create a mechanism that pushes a liquid formulation through. When the needle goes down into the tissue, the drug is already in the tissue.
Mm-hmm. So the next question is also a question I would like to ask. So when do we expect to have human trials and with what kind of drug?
Another interesting question and something that certainly I have been thinking about a lot. I'm very invested in this technology. It's my baby and I would love to see it make as big of a difference as possible. And so right now we're in the process of doing multiple large animal trials, not just in swine, but also in other animals, such as dogs, which are another good surrogate for humans. The estimate is 1 to 2 years at this point, and we have not made a concrete decision as to which molecule is likely to be brought into human trials first. Rani Therapeutics is using octreotide for acromegaly. And the reason they're using octreotide is because it has a very small dose of about 100 micrograms. And so it's very easy to deliver such a tiny dose. But we have not chosen which one we want to deliver for our first human trial.
So the next one also is about dose. Like, is there any predicted upper dose limit of the medication by volume or by weight for like the 8 of these systems?
Yeah, so in the science paper, we were delivering in the hundreds of micrograms limit. So we've seen all the stuff that we've published has been 700 micrograms or less of dose delivery. And that has a little bit to do with the fact that we're delivering insulin and we don't want to deliver much more than 700 micrograms of insulin. We're lucky that the system that we've chosen for the Soma system delivers an extended release time period. So we're not causing hypoglycemia immediately in the swine and causing any sort of damage to the swine in that regard. But in future studies, we've been able to deliver up to 5 milligrams of drug. And that's using different techniques that unfortunately I can't talk about today because they're not public yet.
Mm-hmm. Great. So next is about, I think, is the compliance of this technique. People think that injecting a pill is different from taking a common drug. So will the patient accept this new technology?
Ultimately, that comes down to the patient. There actually hasn't been shown a difference in compliance between taking a pill and taking an injection. What is really the big difference is the ability to get the patient to start on a drug when it's a pill versus when it's an injection. So patients have been shown and physicians have been shown to delay insulin initiation about 7.7 years after a patient should begin taking insulin. And a large percentage of that is due to the fact that it is an injection. So oftentimes the first line of defense for diabetes will be a small molecule drug that's dosed via a pill just because it is a pill and patients feel like they're healthier when they're able to take a pill. So that's really where the greatest benefit can be seen, is hopefully getting patients to take these macromolecule drugs earlier in the onset of their disease rather than having to wait a longer period of time.
Mm-hmm. So the next one is about the FDA? Has the FDA provided any guidance to the size of phase 1 or phase 2 trials?
So the best example of this, I guess, would be the Rani Therapeutics clinical trial, and that one was not done in the U.S. It was done in Australia, and they dosed about 40 patients with the device that they were using that targeted the small intestine, and then they had, I think, somewhere between 5 and 10 people who were controls, and so I think that that would probably be a good size for phase 1. But that being said, they did it in Australia. They didn't do it in the US. And so the size might be a smaller or larger size. There might need to be a proof of concept study as well that's done before a phase 1 trial is done.
Mm-hmm. I think the next one is still about the dosage, like practically speaking, the need for short-term insulin has widely varying dosage. So is there any like a way to address this?
The need for short-term insulin does have an extremely varying dosage, and I don't think that short-term insulin is going to be the best go-to for this particular pill. I think it may be necessary to still take injections for short-term insulin. But that being said, as we mentioned before, this device can load different amounts of drug very precisely, and because of that, you could have different pills with different dose sizes.
Mm-hmm. I think the next question is about the fabrication. How are these microneedles are fabricated, and how constant is the dosage applied to these needles?
So when we were fabricating the needles in-house, we used a pill press, and so it's basically just a compression of the insulin mixture or the drug mixture, and then on top of that is just the polymer mixture. And so it's just one simple press. It's made exactly how you would make a tablet, and it's using the exact same system, just a slightly different mold to create the post shape. And the variability that we saw between the doses that we were creating was about 5% in that initial study. But that being said, this was done in a lab, right? It wasn't done using a machine. It was done by hand.
Mm-hmm. So the next is, would this be considered as a combination product, a medical device plus a drug, or is just a drug?
It would likely be considered a drug device combination. And additionally, because you're delivering using a different method, a different space in the body, likely it would be required that every new drug that was brought to market using a system like this would need to go through a clinical trial.
Okay, great. I think we are running out of time, and I also got a few comments that saying thank you and a great talk. So a reminder to all viewers, you can ask questions using the Q&A feature in the Zoom, or you can comment on the YouTube live. So on behalf of the Alumni Association, I thank you for tuning this faculty forum online, and a big thanks to Dr. Alex Abramson. And the alumni office staff will be sure to forward all the questions. I think some of the questions were not able to be addressed in air. And also, you can tweet about today's chat using the hashtag MIT Better Work. And also, you can send following up questions and feedback to alumnilearn@mit.edu. And thank you again for watching. Thank you.
Yeah, thank you all. Thanks for watching.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.